Hi, welcome to the third Thought Leader Thursday. I'm Melissa Figgins, and I'm the Executive Director of Positive Outcomes with Quality Executive Partners. We are a consulting and technology firm focused on delivering innovative solutions for the life sciences industry. In 2019, we launched Virtuosi, which is transforming learning in the pharmaceutical industry. It combines video and VR interactive experiences in a full 80-hour curriculum that's focused on aseptic manufacturing and microbiology. I'm so excited to lead today's webinar and especially to introduce our two thought leaders. Let's bring out Bob Farrer first.
Hi, Melissa.
Hey, Bob. Bob is the Vice President of Consulting Operations here at QXP. Hey, Bob, if there were a few people maybe on the line probably who didn't join the first Thought Leader Thursday when you presented, maybe you can give a little bit of background on your experience and your passion for teaching.
Sure. Well, thanks for having me back. You know that I love doing these things as much as you do. I've been in the pharmaceutical industry for a little over 30 years at this point. And originally, when I went to university, graduated with a degree in engineering chemistry and thought I wanted to be a doctor. But when I realized that I didn't have the passion for going to school for another 8, 9 years, I decided to go into making drugs, of course, ethical pharmaceuticals. And I've been doing that ever since, working in the pharmaceutical and biotech industry. What really drives me and what my passion is education and learning. When I first started in the industry, I had the good fortune to work with people who were my mentors, who knew the science behind the compliance, who understood why we were doing what we were doing. So I wasn't just trained on an SOP to do a task, to follow rote instructions. I really got the scientific understanding of the operations and the processes that you didn't get at the university level. So for me, what I want to do is, now that I've spent 30 years in the industry, is I want to give back to the younger people, to the people who now are coming into the industry, and really help them become successful.
That's awesome. Wow, you are totally a thought leader, and I'm so happy to have you here. It's going to be a
Oh,
great,
thanks.
great webinar. Let's bring out our second thought leader, Becky Brewer.
Hi there. I'm Becky Brewer, and I also have 30 years industry experience, and I am an instructor for ISPE and PDA. Obviously, I'm also one of the participants in the development of Virtuosi, and I really also have a passion for teaching and a passion for talking about pharmaceutical manufacturing, including aseptic processing. When early in my career, I had the opportunity to participate in the validation of an automated particle inspection system, and that really spurred my lifelong interest in the inspection of pharmaceutical products for particulate. So I'm excited to be able to talk today and answer some questions for some folks.
Wow, awesome. Yeah, I mean, this dynamic duo here, they're amazing people and they're changing the pharmaceutical industry, making the life sciences just, more accessible through their experience, their training, and their passion. And this is exactly why we started Thought Leader Thursday, is to give you access to people like Becky and Bob, and also to bring into focus these important topics like visual inspection. Now, during this webinar, we're also going to give us uh, uh, provide some about them, but showing a little bit about like how you would do that. So you'll go to the top of your screen and select that orange arrow button, and then you can, that opens up your menu window. Click on the question window, and you can just type your questions in and ask your question anytime during the webinar. So you don't have to wait until the end when we're in that actual Q&A session. But before we go any further, I'd like to just sort of I guess get right into visual inspection. But in particular, I guess I'd like to see if we could also talk about why. I know we're going to go through some videos that help us understand the requirements for visual inspection, and I know Bob, you're going to show us a little bit about how the virtual environment works, and you can even show us how to do a proper visual inspection. But I want to get to the essence of this topic and why visual inspection is important. And I think, Becky, this is something that I know you're really passionate about. Maybe you
Yeah.
can explain why visual inspection matters so much.
Yeah, well, thanks, Melissa. All Virtuosi episodes really focus on the harm to the patient and how we can prevent that through the actions that we take in the environment. And so in this particular episode, we talk a lot about how particulate can harm patients, the different types of particulate, the reasons that particulate should be prohibited from entering product as a primary source, and then when that fails, to ensure that we have a robust process on the back end to screen the product to ensure that the particulate has been removed from the resultant product lot. Now, this episode also focuses on how to do that robust inspection correctly. And the why really comes out when we discuss why we inspect product in front of white and black backgrounds, why we inspect product for a minimum period of time, why we must invert the containers before we inspect them, why we must swirl them to ensure that we set the contents in motion before we provide that inspection. In this episode, we primarily describe the manual visual inspection from a technique perspective, but we also talk about how that is important and relates to both semi-automated and automated inspection methods. The manual inspection is the foundation for how we validate those other two system types and how we ensure that those system types work effectively. So without delaying this any further, let's get started with the first video. But let me also mention that in this episode, we also talk about integrity of containers. And so it's important that the integrity topic is also mentioned at the beginning, and that's because that's another part of the episode that we're not going to be showing today.
Okay, let's cue that video.
In addition to container closure integrity testing, visual inspection programs are performed to look for visible particulate matter in all sterile injectable dosage forms, defects in assembly and sealing, and cosmetic defects. These inspections will also look for other failures, such as visual appearance defects in lyophilized products and fill volume errors. Inspecting for particulate matter in an injectable product is important to protect patient safety. Particulate within an injectable dose is considered contamination. It could be from a microbiological or non-microbiological source. This contamination could cause infection, clog a blood vessel, or lodge within an organ and present a health problem for the patient. An integral unit that passed a container closure integrity test may have an observable assembly defect, such as an improperly crimped overseal or a failure at the interface between the needle and the syringe barrel. These could indicate a future premature integrity failure. Cosmetic defects are not directly related to product squips. However, if a statistically significant number of cosmetic defects are called from a batch, when compared to routine reject limits, this could indicate a more serious manufacturing problem which might require investigation. The primary focus of this segment will be on visible particulate contamination. Regulatory documents indicate that articles intended for parenteral administration are manufactured in a method designed to exclude particulate matter. However, it's a requirement that each final container of parenteral product be inspected to the extent possible for the presence of observable foreign and particulate matter. Inspection processes must be designed and qualified. Containers which show evidence of visible particulate shall be rejected. Every parenteral product lot must be essentially free from visible particulates. The term essentially free means that when injectable drug products are inspected as described in the United States Pharmacopoeia, no more than the specified number of units may be observed to contain visible particulates. The general requirements of a visual inspection program can be summarized as follows, with the following 5 general topics. particulate identification, prevention, and continuous improvement, ability to detect particulate and other defects, inspector qualification, defect classification, and AQL sampling program. It's improbable that any inspection process can reliably detect 100% of defects. If a process produces too many defects, it reduces the amount of product that's available for the patient. Therefore, prevention of defects must be the focus of any manufacturing operation. All component design, procurement, receiving, processing, filling, and sealing steps must be carefully established and, where appropriate, qualified and validated. These steps are designed to prevent defects and minimize particulates from entering the product. There are 3 types of particulate contamination that can occur within a dosage form. Particles may be extrinsic, intrinsic, or inherent. Each of the particulate types has a different risk associated with it, and each must have a prevention and control strategy. Extrinsic particles come from somewhere outside the process. This could be particles such as hair, fibers, rust, and insects. These pose the highest risk because it's unknown where they came from and how they got into the process. These particles tend to occur randomly, so finding the source and preventing a reoccurrence can prove challenging. Also objectionable and potentially harmful to the patient, intrinsic particles are pieces of material from within the process. Some examples of intrinsic particles include approved product contact materials such as pieces of gaskets, stainless steel, and glass. When found in a sterile injectable dosage form, both extrinsic and intrinsic particles render a unit unacceptable, and the unit will be removed from the batch. Both extrinsic and intrinsic particles should be categorized, analyzed, and trended. If the occurrence rate of either particle type increases, this could indicate a larger contaminating event. An investigation should be performed to determine root cause. If the investigation finds the source of the problem, corrective actions can be implemented. The final type of particle is known as an inherent particle. These are particles that are part of the formulation, and they form through clumping or agglomeration. The criteria for acceptance or rejection of inherent particles observed during visual inspection depends upon the requirements stated in an individual regulatory filing, a national testing monograph, or company procedure. As part of a particulate and defect prevention and continuous improvement program, it's imperative that the defects are characterized and analyzed to determine how the defects occurred and how they can be prevented. Companies employ many different process improvement tools to achieve continuous improvement goals. For Virtuosi, the tool we developed is called ICE ACR. This acronym means identify, characterize, evaluate, analyze, correct, and regulate. Which tool to use and how to use it is highly dependent on each company's philosophical approach to problem solving. The important thing to understand is that before you can improve a process, the problem must be identified, characterized, evaluated, and analyzed.
Wow, that gave such a great overview of what, types of things you need to think about when you're creating your program. Now, I know in addition to video, you also created a VR experience. Can you, Becky or Bob, talk about a little bit what you had to consider when you developed that virtual experience?
Yeah, I don't want to hog all the time here, but I would like to talk a little bit about the development because it was really an interesting part of developing the VR for Virtuosi. I think that one of the big challenges was how to make visual inspection realistic within the headset, how to ensure that the requirements were clearly visible. We could see the particles in the headset within the different confines of people's visual acuity and also within the confines of the pixelation that naturally occurs within the headset, but not to make them so large that they might appear cartoonish. We wanted there to be some realism associated with it. We discussed various defects at the beginning of the virtual reality activity. So we talked about the particulate that you might see and the different types. We performed the inspection on 3 different container types. So we performed the inspection on vials, ampules, and IV bags. And today we're going to do the ampule section, or show you a piece of that ampule section. In the Virtuosi activity, there are 10 of each container type that are inspected, and there are about 7 containers with defects, which is a high number of defects for that population. The intent here was not to try to mimic a qualification library of defects, but really to ensure that people had the opportunity to see the different types, to experience the requirements of why we look in front of white and black backgrounds without making that process overburdensome within the headset. Again, our intent was not to replace on-the-job training, but simply to provide that foundational baseline for people to ensure that they all had the same level of training and experience before, to prepare them for that OJT activity. In Virtuosi, one of the activities that we also require is that they learn to categorize the defects that they see. In this way, we can ensure that the activity is more than just guesswork on the part of the inspector, but that they're actually learning to differentiate and understand what kind of defects they might encounter in the real world. As with all Virtuosi VR, the activity provides feedback, and so there's real-time coaching that occurs when the inspection is done incorrectly. And this is part of what you'll get to experience in the video that we're about to show you for that. Now, Bob, I think you did this video, so why don't you talk a little bit about how you prepared the video and what you did for it?
Yeah, so the video that I'm going to show is actually me performing a visual inspection. And I think, Becky, I only did 9 of the 10. The first one I was kind of warming up to make sure that everything was going to be focused for people, because remember, you know, what I see in the headset is a much larger view, much wider field of view. Than what you're going to see in a recording. So what I did was I set up a screen capture to record what I was seeing, and now we're going to stream that through the webinar so that everybody else can see it. I recommend that everybody make sure that your screen, the video window is maximized so that you can see the particulate. And as I'm actually going through the inspection, I'll talk in real time a little bit to point out some things that you should be looking at, just to make sure that the image quality that you're seeing is as good as what I saw in the headset. You know,
That's great.
yeah, Becky, you may remember when we were developing this, how we had to go through over and over and over, you know, doing the visual inspection. And I tell you, even though it's virtual reality, I got real problems with my shoulder from it, from doing it so much. So I really have a newfound respect for people who do manual visual inspection day in and day out.
Yeah.
Now there's no audio track over this, and I'll point out in advance, I did deliberately make one error so that we can see what the feedback is. But I have the audio off during the recording so that I can narrate what we're doing. So Melissa, maybe you can take it from here and get the video queued up.
Yeah, let's get into this video. And just a note for everybody is you might get a little bit of delay as we go in and out, but we'll see you back here after the video.
Okay, so you can see here that I've grabbed my first ampule, and in Virtuosi we invert and we swirl. Some companies choose to only invert their units to move the liquid. And as you can see, I was able to see a particulate. I now categorize what the particulate is. It was light fibers, and because I've detected a defective unit, I can cease the inspection immediately. Had I not seen a defect, I would have to complete the inspection in front of the light and the dark, or the dark and the light background, and the order of the two doesn't make a difference. Now, as I mentioned, in the headset, these particles are visible, but you can see that I'm leaning in quite far to ensure that the recorded image allows you to see those dark fibers in that case. And now we'll select the type of defect. But normally, if you were doing the visual inspection, you would do it at a comfortable arm distance, just as if you would do it in a manual inspection booth. You wouldn't need to lean in the way I'm doing it, just to ensure that the recording is acceptable. In this one, you can see small air bubbles rising. And just like in the real world, we need to make sure that we give that air bubble enough time to move to determine whether it's an air bubble or whether it's some sort of a particulate defect. And again, we can see small air bubbles rising, but in this case, there was no defect. Therefore, I put it into the right-hand side, which in this particular setup is my acceptable units. The way we've set up the inspection booth is to ensure that we don't have movement of good and bad in the same direction. So you'll see I pick up a unit from the left-hand side, which is uninspected. If it's a rejected unit, I also put it back to the left-hand side in a separate tray. In this case, again, it's an acceptable unit. Therefore, I put it into the right-hand side tray, which is the acceptable units. You may notice off to the right side, as I'm putting it into the acceptable tray, there's a small placard that we have in virtual reality. And what that placard does is it allows you to repeat the tutorial portion of this if for some reason you've forgotten a proper step, or if you're unsure of why you need to do something, you always have the ability to go back and rewatch that portion of the demo. Let's see, in this unit, we have to determine if those are air bubbles or not. Well, I see some things that aren't floating, so they're not air bubbles. And in this case, they were glass particles. So in Virtuosi, glass particles and stainless steel particles look very similar, just as they would in real life, and they're difficult to determine. And we deliberately tried to make it as realistic as possible. Now for this one, I'm going to deliberately select the wrong answer so that the system will tell me what I've done or what I've not done correctly.
Sorry, that is incorrect. Repeat the inspection to correctly identify the defect.
So now I'll reclaim the unit that I incorrectly classified, and I will re-inspect it. And as I mentioned a moment ago, if I was unsure what I was doing incorrectly, I could go back and rewatch the demonstration. This time I'll categorize it correctly because I'll try not to make the same mistake twice. And in this unit, we see some air bubbles rising, but some particles are not rising, so therefore it's a defect, and we categorize those defects, and our inspection is complete.
Oh, that was awesome. I mean, I can totally see how just even telling the difference between an air bubble or, an actual particulate requires a lot of attention and patience. It's really cool.
Yeah, it certainly does.
Now, I know you have another video that just to kind of, pull this education piece together. Maybe you want to talk to us about what we're going to see in that final video, Bob.
Yeah, sure. So, as Becky said, the entire course includes two different subject matters, and we're showing a little bit about the visual inspection. So I don't think that we have time, Melissa, to really cover the entire next segment that I was wanting to show. But what we are going to talk about is the human factors that are related to manual visual inspection. So in the first part of this video, it's going to discuss topics such as visual acuity, the importance of proper lighting, viewing distance, rate of inspection, all of those things that directly impact the manual visual inspection process. Unfortunately, the portion of the video that we won't show today discusses other human factors which are equally as important, sometimes more important, and that's how we would minimize distractions and maximize comfort for the inspectors. So we tend to want to have manual visual inspection done in a quiet area. We don't want people to be distracted with people coming and going and walking past in the hallway and waving, as they're going by the window. And we also want the inspectors to be as comfortable as possible, because as you can imagine, sitting in one location, concentrating on units, for hours and hours on end as you're doing this process, is not only tiresome for the person, but it's tiresome for the body. I tell you, until I started working at home due to the current situation that we're all dealing with, I thought my desk chair was just fine. I'd spend, an hour, 2 hours, whatever, working. And now I'm telling you, I can't sit still for more than, 20, 30 minutes without fidgeting and having to get up and move around. So for someone who's doing manual visual inspection, comfort is really one of the most important things that can be done. So I think that, if we're ready, we can cue up the video, Melissa, and I'll let you
absolutely.
take it. Yeah.
Okay. Yep. Here we go with the video.
Researchers have found that a person with normal vision, sometimes referred to as 20/20 vision, can see an object as small as 100 microns at a distance of about 2 feet. 100 microns is about the width of a human hair. This threshold is assuming that the inspection is under adequate lighting conditions and the item is in the center field of vision. As light decreases, objects are more difficult to see. Likewise, the ability to see an object decreases as it approaches peripheral vision, or vision away from the center of the field of view. Therefore, under the very best case conditions, 100 microns is a theoretical maximum sensitivity of the human eye with normal vision. Research highlights several factors that affect the ability to identify and remove units that contain particulate and other defects. Inspection effectiveness is highly dependent upon procedures, technique, and working environment, properties of the product and container, the workspace, and human factors. Each needs to be considered to optimize the effectiveness of visual inspection by humans. Procedures and technique include subjects such as lighting, background, viewing distance, rate of inspection, and motion of the container contents. The lighting level must be sufficiently bright to allow the eye to see defects. However, too much light causes eye strain. The environmental background lighting of the room should be low, so there's good contrast at the inspection station, and so that environmental light is not casting shadows. The inspection lighting should not flicker, as can occur with some poorly designed fluorescent bulbs. Lighting must be positioned so that there's no glare, and it should be directed at the container under inspection and shielded from the inspector's eyes. For manual visual inspection, the major regulatory agencies, as well as national pharmacopoeias, have aligned that a diffuse light intensity between 2,000 and 3,750 lux at the point of inspection should be used. To visualize this, 2,000 lux is the approximate brightness of a typical overcast day at noon. Depending upon the type of container being inspected, such as an opaque plastic container or an amber glass container, a different light intensity might be required. This should be justified and included in the process qualification. Specialized lighting, such as Tyndall lighting, could be useful when inspecting glass containers for defects. Tyndall lighting is a type of light source that is introduced at a right angle to the container. This lighting will cause internal refraction of defects in the glass wall, as well as reflection of particulate within the container, if it's present. When containers are inspected for defects, and especially particulate, the contrast of the background is important. Dark particles are usually more visible against a light background, and light particles are more visible against a dark background. It's important that the background not be reflective or produce a glare which could impact the inspector's ability to see potential defects. Harmonized regulatory guidance for manual visual inspection states a black and white background should be used. The viewing distance of the container being inspected is critical for a couple of reasons. As previously stated, someone with normal vision must perform visual inspection. Corrective lenses may be used to achieve normal vision. However, the distance at which the inspector has normal vision may vary. Someone may need corrective lenses to see distant objects, while others may need corrective lenses to see near objects. The inspection distance must be constant, approximately 2 feet, so that the corrective lenses worn are optimal for the given inspection distance. It's for this reason that the use of multifocal point corrective lenses, such as bifocals or progressive lenses, should not be permitted for visual inspection. In addition, viewing the container at a consistent distance avoids a need for the eyes to refocus, thereby reducing eye fatigue. To further reduce operator eye fatigue, it's best practice that operators receive frequent breaks from performing visual inspection throughout the shift. Regulatory guidance doesn't specify that magnification is required for visual inspection. However, it can be helpful. Keep in mind, distortion near the edges of the lens can occur, so if magnification is used, care must be taken on how it's implemented. The rate of inspection is an important factor to ensure an operator's ability to detect and remove defects. If the container is viewed for too short a period of time, the eyes won't have time to scan it and its contents. Harmonized regulatory guidance for manual visual inspection states that a 5-second inspection against each of the 2 different backgrounds should be performed. The human eye is more adept at seeing movement than it is at seeing a stationary object. A container is gently inverted and returned to its upright position. This ensures that liquid has contacted all interior surfaces of the container, which would ensure the particulate would be in the solution. Then, the container is gently swirled to put the particles into motion, since particles in motion are easier to see. Particles are observed, and watching whether a particle floats or sinks helps the inspector identify the difference between a small air bubble, which will rise, and a real particle. Care must be taken to not agitate the container, as the formation of bubbles could cause interference with identification of actual particles. In addition, if your company's procedures allow or require that more than one container is inspected at a time, care should be taken so that portions of the containers are not obscured by the hands, which could mask defects or create blind spots. Specialized holding tools for inspection may be useful to minimize blind spots.
Hey, during that segment, we heard that operators should receive frequent breaks from performing visual inspection throughout their shift. That sounds like a great question to poll the audience with. Let's see
I
if we can.
agree.
Let's put that poll out to the audience and see what their experience is.
How are we doing on the answers, Melissa?
They're coming in. Looks like we have, I think 5 minutes every 30 minutes is winning. 5 minutes every 60 seconds is tied with more frequent than every 30 minutes. Let's let it go just another maybe 10 seconds, and then we can display for everybody. But definitely it's 5 minutes every 30 minutes is winning for sure. It's about half our group. We'll make sure that everybody can see the results of this poll though.
So is the poll closed?
Closing it now.
Great.
There we go. So you can see it on your screen here. Bob, is this something that surprises you, or Becky?
No, it doesn't surprise me at all, Melissa. Right now there's really no harmonized guidance, although USP 1790 does suggest taking an eye break at least every hour to rest the eyes and mind. And then it describes a short break, and it describes short as 5 minutes, or it goes on to say, or a longer break such as a lunch break. So 5 minutes every 30 minutes doesn't surprise me at all. Just remember, break times have to be enforced. And they have to be documented. And each company really needs to define what that standardized period is to rest your eyes and to give the operators a chance to, let's say, recover, because it is a very stressful type of process if you haven't done this before. One of the things that I realized in one of the last companies I was at, Melissa, is that they were struggling, how to document and record this appropriate amount of time. And what they wound up doing was they realized that inspecting a tray of product took about 35 to 45 minutes. And so what they decided to do is that after every tray of product, each inspector took their eye break, and then they documented that as part of their inspection start-stop time, right? And that worked out very well for them. While another company that I was at recently, what they had is they had a supervisor who would announce when it was time to take the break, and that supervisor then recorded the break time for everybody. And during that time period, all of the operators stopped what they were doing and closed their eyes and rested for a few minutes. So I think both of those are viable options in ways to let's say, ensure that the breaks are not only taken, but that they're documented properly. One thing that I would not recommend, however, is to allow each person to monitor the time and then, let's say, self-take the break. And the reason is because, if I'm doing a visual inspection and I know that I have to take my break because it's a requirement, I'm going to be looking up at the clock and I'm not going to be focusing on what I'm doing because I don't want to be in violation of missing my time. So that would be the one catch that I might, give you as a heads up to say,
Right.
not the best method that I've seen.
Yeah. Wow. That's
Yeah.
awesome. Yeah. Oh, did you have something to add, Becky? Sorry.
I was just going to say that's a great experience, Bob, and a great tip for folks who haven't set up their programs yet.
Yeah, definitely. And this is great discussion, and I love engaging with the audience. That poll was a great way to sort of get that started. But let's start taking some questions and pivot to the Q&A portion. I know this is what a lot of people really wanted to get access to you. And I have a question already pulled up here. I'm gonna let you guys decide who wants to answer it, but I'll read it exactly as it was posted. It says, I've heard you discuss manual visual inspection and touched on automated inspection. My company uses semi-automated inspection. What are some of the issues that we should be aware of as it relates to our visual inspection process?
That's a great question. Hey, Bob, why don't you take that one?
Yeah, I was going to say, so the we saw what manual visual inspection is, right? The operator picks up the unit, puts the liquid into motion, visually inspects it, and then dispositions it. What semi-automated inspection is, for those who may not have this technology in their facility, is the material handling of the units to be inspected is done by a machine. So they come in, they're loaded onto a machine, they pass my field of view at a set rate, a controlled set rate, and then I have to, the human, determine whether it's an accept or reject. And depending upon the piece of equipment, there's either a signal button that I press, or I reach in, stop the unit, reach in, remove the unit, etc. So the things to remember is that I'm still the detection device, right? The machine is just a material handling portion of the process. So some things to look out for are making sure that the speed of the machine is always set at or below what I've qualified at. And typically, a company will qualify everybody at the same rate of speed so that we don't have this issue of a fast machine or a slow machine. The machine itself has to be sure that we're maintaining it properly, and the reason why that's so important is not just from an operational perspective, but let's say the mechanism that puts the solution into motion as it's rolling, usually it's some sort of a rubber gasket that'll turn the unit. If that's not working properly, then I'm not going to rotate the unit. I won't see all sides of the unit. The particles won't be in motion. It just becomes, very problematic and not a valid inspection, if you will. Another thing to think about is any type of defects that could occur in my field of view, whether it's on the rollers, whether it's on the background, or if there's some sort of let's say a plexiglass shield. I've seen this sometimes over the units to either protect from breakage or to make sure that they're held in place. Any type of scratches, marring, defects, all of those things now make it difficult, if not impossible, to determine whether the defect is in the unit or in the machinery or the componentry of the machine. So I would say that those are just a couple of the things, without rambling on for the whole time, those are just a couple of the things to look out for.
Yeah. I think that's great. Let me get to another one, and maybe Becky, this one's a good for you.
Okay.
Good one for you. It says, or it states, are agglomerates or inherent particles considered defects in visual inspection?
Yeah, so the compendial standard for products says that we need to be essentially free from particles. And for a biologic, for example, proteins tend to agglomerate, which means that they form small clumps. And these clumps form what becomes a visible particle within the solution. It's fairly typical that either the monograph for the product or the regulatory filing for the particular product will have a limit as to how many agglomerates are considered acceptable or typical of that particular product. And so it's very important to adhere to your company's standards for that. If you see a batch where there are more agglomerates than typical, that could signal a processing problem, such as some shear force or some unusual temperature that the product might have been exposed to. So it's really important that these be identified as outlier issues and that those batches be segregated and inspected. Since agglomerates can cause an immunogenicity problem, it's very important that we control these for patients. But again, it's so product specific. It's really hard to provide a guidance that's appropriate to everybody.
Yeah. Let me go to another one here that's just gotten submitted. I'll read it exactly as submitted so that I don't take any important parts out. This one states, how are limits established for critical defects during visual inspection? At what point do I need, or do I know or need to investigate a high number of critical defects? I don't know if Bob
I
would
can take this one
would
if you
like
want,
that one.
Becky.
Sure.
Let me just make sure that I understood, and I'll answer the question as best as I understood it. So first of all, critical defects, major defects, minor defects are the typical classifications that we use, right? And inspection is a probabilistic process. There's no assurance that we're going to get 100% of the defects out of a batch. Now, the goal, of course, is 100%. The goal is, identify every defect. But the reality is that the limit of detection of the eye, as we discussed in the episode, there is a finite limit below which a defect may not be seen by everybody. And all three of us are wearing corrective lenses. Even though our eye vision is 20/20, I may be able to see a particle that's 120 micron, and Becky may be able to see a particle that's 100 micron. So without rambling and trying to get back to the question at hand, the issue is that I want to remove all of the defects. Now, in my AQL, which is the requirement to then do a QA inspection of the 100% inspected products, I set the limits for critical defects very tightly, maybe a 0.1% according to the ANSI standards. And there it says that if during that AQL I find 0 major defects, and we're only talking about, I'm sorry, critical defects. I misspoke there for a second. But if I find 0 in my AQL, then the batch is considered passing. If I find 1, according to that particular standard at that inspection tolerance, 1 found during the AQL fails. So I think that this is the question that the person was asking about. How do I set those limits? I just want to just one more brief point, Melissa, is that when we qualify inspectors, many times to ensure that we pay the most attention to the most critical items, we tend to set a requirement internally for our process that an inspector must remove 100% of the critical defects in the test set in order to be considered passing. And as I just pointed out, it's probabilistic. So ideally, they should be able to get 100%, and maybe that's how your qualification program is set up. But remember, what we're really talking about is that AQL process that releases the batch.
Right. Okay. I've got actually a question here, and it has to do with how we use VR. And so it says, could
Notice the timer.
VR, Yeah,
Yeah, Bob uses it
exactly.
right in the same room he takes his webcams videos.
You are dedicated. This one says, could the VR also mimic viscosity of different solutions in the vials?
Absolutely. I'll take that question. So when we created the VR activity, we created it to provide a swirl and a relative viscosity so that the air bubbles took some time to move to the top of the solution, so that a person would really be focusing on observing the behavior of particles in that solution. So we established it based on sort of the everyman's solution in the vial, but we know that different viscosities will in fact slow down the motion of particles, and that's certainly possible to do in VR. In our particular environment, I would say that we've created all three solutions to behave about the same within the constraints, but that certainly is a possibility within VR to slow that down, slow that motion down, slow those particles down. And that's typically what happens. Viscous solutions can also cause some problems in getting particles suspended, especially if they're stuck to the edges of the container or the walls of the container. So that could also be easily simulated within VR.
Okay, that makes sense. Bob, I think this might be a good one for you. So it says, which is the general expectation for frequency of verification for light intensity, lux, at the manual inspection station? Every time before the inspection, every quarter, every year?
Yeah, so I wish that there was a simple, simple answer that you just say, this is what it is. Depending upon your light source, bulbs do not always emit the same amount of light over their entire lifetime. So if we start from that premise, then we have to determine how does the light degrade over time based upon the light source. If it's a fluorescent tube, if it's a halogen light, or if it's an LED type of bulb. We know LED bulbs now are the most common, or I shouldn't say the most common, the most popular bulbs being sold, and they tend to have a very long lifespan, and they would tend to then hold their light intensity for a longer period of time. So giving the why or the science behind the answer first, I would say that based upon the type of light source that you have and the knowledge that you have in your facility of how these light sources degrade or don't degrade over time, I would set that interval of checking based upon that. And this is something that you can do as part of, let's say, a protocol to establish, monitor it once a week for a period of time and determine if there's any degradation, then put it onto a schedule such as calibration. On the other side of the equation, quite honestly, it's a fairly simple test. It's a handheld meter that you hold at a fixed location. You determine, what the light is, and you document it. So it's not that onerous. If a company chose to just say, I'm not gonna take a chance. I'm gonna record it before I start, my daily shift, right? So again, from one side to the other side.
Yeah,
Right.
but important, right? Just to make sure that you
Absolutely.
have a
Absolutely.
plan and it's documented, and yeah,
Yes.
that makes great sense.
Mhm.
Here's a great one. What is the ideal method for visual inspection for product in a dull container where vision is not easy?
Ooh.
Yeah. Do you want?
Yeah,
We
well,
can both take that. Do
it
you want to
was
take that, Bob?
juicy. No, I'll get ahead. I'm rambling. I'll let you have a turn.
Okay, I can ramble now. Yeah, so this is a challenging topic, and there's a wide range when we talk about difficult to inspect containers, right? Because we could have something that's fully transparent, translucent, or truly amber, or almost all the way to opaque, right? The compendia provide some guidance. The new 1790 talks a little bit about this and about the fact that, hey, we may not be able to fully inspect some of these truly darkened containers or containers that are difficult to inspect, and we might have to use some withdrawal methods to be able to examine it. But that's a destructive test, and we're never going to do that for 100% of the product. So we need to try to do our best to perform an inspection, whether that means increasing lighting or changing lighting intensity and justifying that based on the container type we have, whether that means increasing inspection interval to ensure that we have a longer time to try to identify materials within those containers. But it's really probably going to come back to offline destructive test methods for a percentage of the batch to identify that that we don't have a particulate problem. Now, even when we can't fully or inspect the contents of the container, we may still have to look at the container closure, the condition of the container, all of those other physical defects that could be associated with the delivery system that we need to ensure are checked for during the inspection process. So
Yeah,
we'll probably never get rid of the inspection process for those.
Melissa, can I add one thing to that?
Okay.
Becky, I thought that was a great answer. And what I just want to remind everybody is that you can't inspect quality into a process, right? So you
Absolutely.
can't. And Becky, to your point of what you were saying about how difficult it is to inspect an opaque container or a polypropylene container or an amber container, this is one of the foundational reasons of why preventing particles in the manufacturing process in the first place is so critical. Because as I mentioned before, it's probabilistic to begin with, and if it's difficult to inspect units, it's bad practice to just let things happen. I mean, we have to keep particles out of the manufacturing process.
And that's what I really liked about the first video that we saw, that the focus was not only on instructing people about the particulate types that we might see, but really the emphasis on prevention, because
Right.
I think the only way that we can ensure that we're providing safe product to patients is through prevention, not through inspection.
Right.
Okay. Here's a good one. I'm not going to decide who who should take it.
We'll arm wrestle a bit.
I'll let you two fight over it. Is there a performance qualification associated with visual inspection? If so, what or how is that done?
Instead of arm wrestle, Becky, do you want to do a thumb war?
No. Do you want to take this one, or I can?
Yeah,
Either way.
I'll take this one. So when we talk about a performance qualification, I think we really need to discuss, first of all, which inspection we're talking about: manual inspection, semi-automated inspection, or automated inspection. So I'll take the easy one first. For automated inspection, there absolutely is a PQ. There's a performance qualification, as part of that. I would say for manual and semi-automated inspection techniques, where I am the detector, right, the detection technology, in both of those situations, there would be an IQ/OQ on the inspection equipment, whether it's mechanical or whether it's just the booth with the light and the backgrounds. But the detection equipment does get a PQ, and that's called operator qualification. And Becky alluded to this before when she was describing how in Virtuosi we set up that there were 10 vials with 7 defects, and she said that that's way more defects than you would normally have in a test set. But operator qualification is performed using test sets of a company's actual filled product, seeded with defects from the actual process where possible, or created to simulate a defect in a unit if necessary. And then the qualification occurs by ensuring that an operator has the ability to remove the defects at a certain rate. And if you remember a moment ago when I was talking about that AQL issue, we set up that qualification process sometimes to say how many critical, how many major, how many minor am I allowed to miss that are defective, but we also have to have a requirement that says that I don't remove too many good units, because that means that I may not be doing my job very well if I'm just randomly pulling stuff out and hoping that I'm going to capture defects.
Randomly guessing whether stuff is bad or not.
Exactly.
Absolutely. Yeah. So there are very strict criteria for how we qualify people. And again, Bob mentioned some of the important characteristics of that operator qualification early on, which included the rate of inspection to ensure that they're operating at the same rate that they would be expected to operate at during routine inspection, and that they have the same opportunity for breaks that they would normally have to have, et cetera. So we really want to make sure that all those conditions are the same in routine production as they were during that qualification.
Yeah, great answers. Hey guys, we are getting so many great questions, but I know we're running close to the hour. So I just wanted to make a suggestion. Would it be okay if we stayed on the line a little bit longer for those who could stay?
I can stay. Yeah.
Yeah, I could stay, sure.
Okay, great. Well, if you have to drop off at the top of the hour because you have another meeting or something, you can contact us at info@virtuosivr.com. You can send a question that you think of a little bit later on that. You can learn more about our product at virtuosivr.com. And lastly, I'll just say before we come back to the Q&A session then that Becky and Bob have just agreed to, thank you, by the way. We have another Thought Leader Thursday planned for next week and for the following. So next week it's going to be microbial limit testing, and just keep an eye out for an invitation for that. But yeah, for those who can stay on the line, let's get back into some more Q&A. I want to make sure that we get through these really amazing questions that are coming in. So I'll go to the next one. This one's interesting. Now, I know it was discussed in video, and we've talked about it a little bit, but let me read it off to you. It says, for manual inspection, the time inspection against black and white background should be validated, 5 seconds each. That's the sort of, I didn't inflect at the end, but that's the question. Should it be validated?
You want to take this one, Becky?
Well, I think it's part of the operator qualification, right? So everybody needs to be trained in the inspection technique, right? They then need to ensure that they can demonstrate that particular technique, and then they need to be double checked in that particular performance of the technique. And so that needs to be something that they demonstrate through their qualification. And during routine performance on the floor, there should be QA on the floor that's helping to ensure that people are abiding by that time. Remember that 5 seconds is really a minimum recommendation, and your product might take more or less time depending on what you can justify for the container type. I think in general, where people don't do less than 5 seconds because of that compendial recommendation, but again, more than that may be required for large containers like IV bags, for example.
Hey, I've got to get this next question then. You just brought it up. So
Okay.
it actually is, does Virtuosi include a module for inspection of large volume containers like IV bags?
Yeah, as I said when I introduced the VR segment, we do 3 inspection activities associated with the VR. We let people take a break because we know that we need a break after every inspection, right? But we do conduct the inspection for vials, ampules, and IV bags, all to teach people a little bit about the differences of what they might see. So in the large volume IV bags, some of the specific education that's provided in the VR is about the challenges and hazards, because not only is the container translucent, it might have printing on it. There are ports off of the bag which have to be inspected, as well as the formation of the bag itself, because there could be defects in the bag or the sealing of that bag. And we also have challenges because the volume is often larger. While sometimes we have IV bags which are very full and rigid, we also have cases where we have IV bags that only have a partial fill because the intent is they get filled with another diluent in the hospital setting. And so those can be floppy and difficult to inspect. And so the characteristics of the individual bag needs to be taken into account when setting those parameters. And so the front of the VR activity talks about all of those risks and challenges to provide the awareness, and again, provide the why behind the inspection, because we want to make sure people are really well equipped to see what they're going to face in the real world.
Yep, absolutely. That's beautiful. This is a great question. It says sometimes we see black particles in the solution. How do you define them as extrinsic, intrinsic, or inherent? Do we need to send them every time to the lab for identification?
I'll take this one, Becky. I
Okay.
would say that yes, you need to identify and characterize the particulate. It could be the source. It could be a gasket that's breaking down. Could be a motor agitator seal that's prematurely wearing and contaminating the product. I guess the question is this is the particulate found before or after final filtration? Certainly after final filtration, it becomes more worrisome. Where these particles are coming from. But typically, a company will have not only a defect library, but they'll categorize what the particles actually are. They'll send them to a laboratory for whether it's microscopic analysis, maybe it's atomic absorption spectroscopy, but they'll find out what that particle is, and
Right.
then they have a way to determine, a potential source, and as we mentioned, eliminate that source.
Exactly. Nice. Okay. Here's one about lyophilized products and just how do you perform visual inspection on those? How do you see the product, or excuse me, how do you see the particles in that product?
Yeah,
Maybe.
let me take that one. I think
Okay.
that one is very much like the opaque discussion we had a moment ago, where it's not going to be possible to 100% inspect a lyophilized product, but it is important to look at the product. And there's a few things that we're looking for. Number one, we'll only see particles on the outside of the cake, right? We'll only see particles that are in the visible range. Things that are in the center of the cake are not going to be visible to us. But there's some other important attributes that we're going to look for when we look at the cake itself. We're going to look at the formation of the cake to see if we have melt back issues, which could signal that there was a problem during sublimation of the batch. We are going to look for whether that cake is well formed or whether it's split, or perhaps there's powder at the neck of the vial, which again could indicate that there were problems during the cycle in controlling the vacuum level. And these are defects that are really important for us to understand because they help to tell us the quality of the product. We still have the container defects we're going to look for. So we're still going to look for stopper placement. We're still going to look for crimp seal. We're going to look for cracks or scuffs on the glass because that's an important aspect of our attributes for the potential failures of container closure integrity. But we're also going to want to take some of that product offline, do destructive testing by reconstituting it, and then looking for particulates in that reconstituted liquid through particle analysis. And again, prevention is key in this case because we're not going to be able to 100% inspect the lyophilized product. And so visual inspection remains an important function, but it's not going to be the only activity that we're going to have to detect particles.
Yeah, there's a kind of similar question popping up here. Let me read this one off, and maybe if I deliver it out, you might be able to just expand a little bit more. It says, what should you do if the liquid in the ampule keeps drying out before the end of incubation and before final day inspection can be done, even with added lids and caps?
This sounds like a media fill question, actually, if they're talking about
Yeah.
final day inspection.
And
So
in an ampule which is fully sealed, there should be no evaporation. So maybe I'll just take it, Becky, and let's make the assumption that they're talking about media fill, and if they're seeing that something is evaporating or drying out, there's a problem with the container closure system. So
absolutely.
this is not a visual inspection problem; it's a container closure problem because a sealed unit, whether it has media or product in it, there should be no evaporation. Now, the only time that I would say that maybe this is a case is if you've got a semi-permeable container, something like polyethylene maybe where you've got some eye drops in it, single dosage. Those are typically packaged in a hermetically sealed foil pouch that has a barrier on it to prevent any type of evaporation. So possibly, I'm just guessing here, the person's talking about these units not being pouched, being left around, and that there's some evaporation before they're able to do the inspection. But I hope between those two answers that I was able to satisfy this person's curiosity.
Yeah, and we can always do a follow-up too.
Yeah.
Yeah, and the only thing I would suggest in that case is see if you can protect the units just as you would in normal protection. So you might choose to overwrap the media-filled containers and then peel down that overwrap for inspection and then rewrap. Of course, you'd have to do a risk assessment on that process, but that's how I would treat it if I was concerned that I'd have evaporation before able to complete the inspection. So again, some ambiguity with the question, but we're happy to follow up with that individual if they need help.
There's some questions about how you calculate your rates. The first one here is, should critical and major defect reject rates be tallied together, or should defects such as cracks and foreign particles have their own specific reject rate or limit? Some companies tally critical and major defects together per defect category.
I'll take this one, Becky.
Okay.
I think that whether you do critical separately and major separately and minor separately, I think that the important thing is that you have to have control of your process. And the reason why we tally these defects and we trend them and we look to see how many we have in a particular classification is so that if we see an increase or if we see a change, we know that there's something going on. So even an increase in minor defects could indicate that there was some sort of a problem, let's say in material handling. So if I see an increase in scuffed vials, for argument's sake, then that might tell me that I had a material handling problem where vials are rubbing together, which could then generate glass particulate. And it might give me a hint that, hey, I might have another problem that I'm not necessarily aware of. So again, the critical issue is just categorizing them and whether you choose to do them together because your particular process generates so few major and minor, major and critical, that it makes sense to combine them. I could see how that would make sense, but I wouldn't say that that's a requirement that you combine them or that it's a requirement that you separate them.
Okay.
Yeah, I would agree exactly with what you said, Bob. And I think looking at them together could make a more stringent criteria for yourself. So that might be helpful for some of your processes, especially when they are very tightly controlled to start with.
Yeah, there's a couple of people who have submitted a similar question, and it has to do with who should be performing the AQL. Should it always be someone in the quality unit? And some are performed by the same person who did the initial 100% inspection. So I don't know what your perspectives are on that.
I'm happy to take that one. So I am of the opinion that it should always be an independent individual performing it. Obviously, that person also needs to be a qualified inspector, so they need to make sure that they've passed the inspection criteria themselves, but it should be an independent person, because if I've already looked through the product and said this product is good, I may be missing a defect type that someone else will catch. As Bob said, this is a probabilistic activity, and so I need to increase the probability that I'll find something by making sure that I've got a fresh set of eyes on the content of that product. Now, in terms of what department they belong to, most companies have it be QA or QA on the plant floor, or have that sort of role or title, but it just needs to be an independent check of the inspection results, and I think that's the important process that's being undergone.
Okay. This one is it has to do with re-inspections also, but let me read it out. It's a little different. Should re-inspections that are performed after an initial failure of the 100% be done under tightened limits or at the same reject limits that the initial inspection was performed under?
Yeah, so most commonly I see companies doing tightened limits for that reinspection. Again, if we've already failed, that tells us that we've got a potential problem within that batch or a problem with the original inspection. And so typically Titan limits are applicable. But again, this could depend on the circumstances of what is being looked for. I've seen cases where people have not used Titan limits when they're looking for a cosmetic defect, for example. So if it was a critical defect, maybe I would require it. If it was a cosmetic defect, maybe I would not tighten the requirements. So it may depend on the circumstances of what the failure was.
Mm-hmm. There's a question about qualification of operators here, and it's should the qualification set used for the qualification of operators contain all types of defects?
Oh, that's a good one. I'll grab this one, Becky.
Okay.
The short answer is yes. We need to make sure that all defects are included in the test set, and not only that, but as we're doing our visual inspection, it is possible that we're going to detect a new type of defect that we hadn't seen before and therefore hadn't included in the test set. So as we're categorizing the defects that we talked about before, we should make sure that if a new type of defect is found, that defect type is then included in future test sets to make sure that operator qualification is comprehensive.
Mm-hmm.
Mm-hmm. Makes sense. Yeah. Here's one. Can you talk and explain about the POD, probability of detection, and related reject zone?
Yeah, I can take that one because, well, first of all, I love to talk, as you guys know. But so the probability of detection really is the way we set up that test set originally. So we take inspectors and we do I wouldn't really call it a full qualification, but we present multiple inspectors with known defects, and we determine what the probability of detection is of a particular defect by having them go through sets over and over and over multiple times till we have a statistically significant sampling to say the probability of all of my inspectors detecting this reject is, let's say, 85%. And I believe that's what 1790 talks about. USP 1790 says 85%. If that's the case, then we use those in our test set. If the probability of detection is too low, it means that it's really at the absolute limit of what someone can see and someone else can't see it. So we tend to exclude those from the test set. So if I remember the question, because now I'm thinking of my answer, I create my test set using the probability of detection. And Melissa, if you can just help me out, I'm forgetting the second part of that question.
Yeah, no problem. Yeah, I'm sorry. And how do you relate it to the reject zone?
Okay, so I think it's basically one and the same, right? So the reject zone, if you will, is this area between, let's say, 100 micron and 150 micron, which is typically where we find that becomes the limit. And then within there, we determine this probability of detection by repeated inspections to then create the test set.
Mm-hmm,
I knew I forgot something. It was the reject zone.
Mm-hmm. This one's a really interesting one. When explaining extrinsic, intrinsic, and inherent, is this only associated to floating particles, or would it also pertain to defects found adhering to stoppers and glass?
Oh, interesting. So, the particulate associated with a stopper could be inherent to the stopper, right? So occasionally we get burned or darkened components from the elastomeric material that comprises our stoppers. And so we could apply it that way. Most companies instead just list that as one of the defects of a stopper failure, a stopper flaw, et cetera, so that they can identify when they've got a problem with the stopper manufacturer or the supplier or that lot of stoppers, because they may choose to quarantine that lot of stoppers and not use them in further product if they detect that there's a consistent problem with the manufacturer of that particular lot. So again, those terms could be applied to defects that are associated with that material, but they don't have to be. We don't have to be so persistent to name everything that we have in that way. It's really more important to determine source, cause, and once we have that root cause, then be able to isolate whether it's something we can exclude from our process.
Here's one I think you guys will really like. It's a great one for discussion. Are fibers always to be considered a critical defect? What if they are found to be cellulose and attributed to some part of the filling process? Could they be considered major defects in this case?
So I think we're confusing potential risk to patient with the type of defect. So as we mentioned before, any visible particulate needs to be removed and rejected. Individual companies set up what they consider to be a critical defect and what they consider to be a major defect. So in one company, you may classify a particle above a certain size as a critical defect, a smaller particle as a major defect. I mean, there's lots of different ways to approach it. So first and foremost, it needs to be removed. I think the question is, if it's cellulose from the filter, is that considered an inherent particle, an intrinsic particle, and if so, does that make it more acceptable? And the answer would be no. Any particle is not acceptable, other than what Becky described being inherent or as part of the formulation. So I would say that it needs to be removed, and whether it's classified as critical or major, I think is secondary.
Okay. I'm gonna sort of mash together two questions because they relate to, again, your ability, viscosity of the solution and color. But I'm just gonna let you know, I think we'll make this our last question. I know we've held the attention of quite a few of our attendees, and certainly Bob and Becky, thank you for your time today. I'll read the two questions just so you can hear it from the how it was posed. But I think you'll probably be able to combine them. So is the process similar for colored solutions, like vitamin B12, for example, where viscosity and color are both problematic? And then the other question that came in, apologies, I'm just scrolling down, there's been a bunch here. Here it is. Do you have a suggestion once the product is inside, for example, polypropylene bottles, and it's in an oil emulsion, so it's not so visible? So in cases like that, maybe you could just give a little bit of guidance and best practice.
Yeah. Again, each material is going to be subject to its own qualification, if you will, of the inspection requirements in order to ensure that you can adequately detect particulate that's in that product. And so you may need to adjust the duration of inspection, the time of inspection that the inspector has to look at the container, or the rotational velocity that you need to apply to agitate the solution to get the contents in motion. For a viscous solution, for example, you may need to influence other aspects like lighting levels or angle of viewing or position of the light relative to the container to try to adapt to different color and opacity risks associated with the product. And so it's really almost going to be a customized inspection based on what works for that particular material.
Yeah, makes sense. Yeah. Hey, this has been excellent. Okay, the questions have been amazing. So everybody on the phone, if we didn't get to yours, we'll come back to you. Or if you think of one, you can submit it to info@virtuosivr.org or .com. But I also hope that you'll join us next week for the micro limits testing, and just absolutely, Bob and Becky did a fantastic job engaging the audience today. This was awesome. Thanks so much.
Thanks.
Thanks.
Thank
It was
you.
a lot of fun. Thank you.
Bye-bye.